So the research in my lab has been focused for the last few decades on how cancer cells respond to certain stresses. And those stresses tend to damage the DNA of these cancer cells, and cancer cells respond differently than other types of cells. And so we have been trying to understand how those differences occur and then how to exploit those for cancer therapy. So the work in our lab has the potential to work with a lot of different therapeutics. One of them is ionizing radiation. So ionizing radiation kills cancer cells by inducing DNA damage. The work that we're doing has implications to try to block how those cells respond to that DNA damage to ultimately make that therapy better. So the work that we're doing under this grant is really a combination of chemistry, biology, cancer biology, and biochemistry. So the chemistry is really crucial in terms of getting it to a point where we can have the experiments work in the laboratory, but then there's a whole other series of experiments that we're doing under this grant to try to make them more clinically applicable agents. We are driven by the data and the sense of discovery. For the vast majority of the experiments that we're doing on a daily basis, they've never been done before, which means the results we get have never been seen before by anyone. And so that is really very intoxicating, seeing a brand new result that nobody else has seen before and trying to interpret that. So it really is the data that comes out on a daily basis. And you look at it at the very microscopic level, right, and a very narrow window of resolution, and it seems like it's just some little result from a single experiment. But when you look at it in the whole, the progress that we can make by having really talented research assistants in the lab, really talented postdocs, generate data on a daily basis, do these experiments, that's what really drives the whole thing. It's the sense of discovery. It's brand new and it's never been seen before. Collaborations for us are really an important part of every project. We are good at a small subset of things in the world, and what we try to do is surround ourselves with people that are good at other things in the world. So we're really pretty well established in the biochemistry arena, but when we get into in vivo therapeutics and how things work in an animal model, we really rely on our collaborators with Karen Pollock over in Vivo Therapeutics Core. Similarly, we're really interested in how cells respond to ionizing radiation, but we're not radiation biologists, so we collaborate with Joe Dinlock and his group to try to understand how our therapeutics work in combination with radiation therapy. So we absolutely could not do this alone. A major player, considering its chemistry, that really drives a lot of this work is a collaboration. This is a multi-PI grant with our chemistry collaborator, Melvin at Wayne State University, who really has transformed the projects by developing these brand new molecules that nobody else has seen before and allowing us to test them and put them into cells, into animal models, and hopefully into patients at some point.